Studies of both mice and humans who have traveled into space reveal that critical parts of a cell's energy production machinery, the mitochondria, can be made dysfunctional due to changes in gravity, radiation exposure and other factors, according to investigators at Georgetown Lombardi Comprehensive. These findings are part of an extensive research effort across many scientific disciplines to look at the health effects of travel into space. The research has implications for future space travel as well as how metabolic changes due to space travel could inform medical science on Earth. The findings appeared November 25, 2020, in Cell and are part of a larger compendium of research into health aspects of space travel that appears concurrently in Cell, Cell Reports, Cell Systems, Patterns, and iScience. My group's research efforts centered around muscle tissue from mice that were sent into space and were compared with analyses by other scientists who studied different mouse tissue, says Evangelia C. Liakis, Ph.D., an associate professor of oncology at Georgetown. Although we each studied different tissue, we all came to the same conclusion, that mitochondrial function was adversely impacted by space travel. In addition to studying the effects of space travel on cellular function, the scientists used a trove of data from decades of NASA human flight expo. They were also able to access data derived from NASA's repository of biospecimens that had flown in space to do further comparisons. Data from NASA's twin study of Mark and Scott Kelly was particularly informative as it allowed for a comparison of the health effects seen in an astronaut in space, Scott, with his Earthbound brother, Mark, who is a retired astronaut. Comparing their studies of mice with human data, Liakis and the team of researchers were able to determine that space travel led to certain metabolic effects. Isolated cells were adversely impacted to a higher degree than whole organs. Changes in the liver were more noticeable than in other organs. This aspect of radiation exposure translates to health outcomes here on Earth for cancer patients who undergo radiotherapy. With this knowledge of radiation's impact on mitochondria, clinicians might tailor radiation therapy in different ways in the future to protect normal tissue. The implications for travel to Mars are especially concerning, the researchers say, as that would involve a much longer time in space and hence lengthy exposure to radiation. The launch of SpaceX earlier this month was very exciting, says Liakis. From this, and other planned ventures to the Moon, and eventually Mars, we hope to learn much more about the effects that spaceflight can have on metabolism and how to potentially mitigate adverse effects for future space travelers. Metabolomics work by Liakis was supported in part by National. Story source: materials provided by Georgetown University Medical Center. Note: content may be edited for style and length. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.